Welcome to today's webinar. My name is Martin Mangold and I'm an application specialist at BMG LabTech. BMG LabTech is a leading global developer and manufacturer of innovative and high-quality microplate readers. Our aim is to provide you with reliable, robust and sensitive microplate readers with made in Germany quality. BMG LabTech are the true microplate reader experts. Since we are the only manufacturer solely focused on microplate readers, have over 30 years of experience in the field, 30% of employees that have been with the company for more than 15 years, an impressive track record of over 10,000 tracked scientific citations in peer-reviewed journals, and high user ratings on independent online platforms. Today it is my pleasure to introduce Martin Schwalm. Martin is a great up-and-coming young scientist with an already very impressive track record of achievements and publications. We are very happy to have him today to show some of his results regarding research on protein degraders. Martin performed his experiments on the Ferrostar FSX, the gold standard plate reader in high-throughput screening. The Ferrostar FSX is the fastest and most sensitive microplate reader. It was specifically conceived to fulfill the hardest screening requirements. It offers fastest detection time, even for dual emission assays, and is the most sensitive reader on the market in fluorescence intensity, fluorescence polarization, time-resolved fluorescence, and TR-FRET applications. This is achieved by assay-dedicated light sources and detectors. The reader is compatible with microplate formats up to 3456 wells and is very automation friendly. The Ferrostar FSX is a multi-mode microplate reader that can be equipped with all commonly used detection modes, ranging from UV-Vis absorbance to alpha screen and alpha laser applications. Since Martin Schwalm performed the experiments he will show today using the luminescence detection mode, I would like to highlight a few features that make the Ferrostar FSX a great luminescence-based microplate reader. First, we have the optic module technology that makes the Ferrostar FSX the easiest reader to optically configure on the market. Optic modules contain assay-specific filters, dichroics, beam splitters, and polarizers, and are optimized by BMG LabTech's application specialists such as myself. While we offer bespoke modules, there is also always the possibility to optimize an optic module according to your specific assay needs. These modules are automatically recognized by the reader via barcode, and you can easily add or replace up to 5 modules in the reader. Second feature is the simultaneous dual emission, which allows to simultaneously detect 2 separate emission wavelengths in one single measurement. This is of course beneficial in ratiometric assays such as FRET, TR-FRET, BRET, and fluorescence polarization applications, and is achieved by 2 hand-matched detector PMTs. For ratiometric assays, the simultaneous dual emission will cut read times in half, eliminate flash-to-flash variation, and corrects for photobleaching, evaporation, and channel decay. One problem that is commonly encountered in luminescence assays is, of course, crosstalk. On the Ferrostar FSX, there are 2 features to reduce crosstalk. For one, we have the physical blocking of stray light by an aperture spoon, as indicated in the figure on the upper right. But we also offer mathematical correction based on a pre-measurement to identify a correction value to address well-to-well crosstalk via the microplate wall. The Ferrostar FSX can be equipped with up to 2 built-in injectors. These are software controlled and have an individual injection volume of 3 to 500 microliters per well, and variable injection speeds up to 420 microliters per second. Reagent backflushing and a low backflush dead volume of under 50 microliters helps to preserve your sample and reduce overall costs. A great feature for very fast assays, such for example flash luminescence, is the ability to simultaneously inject and read at the same time. A new feature available with the Ferrostar FSX is the advanced assay stability. This feature matches room temperature and maintains the reader at any room temperature between 18 to 45 degrees Celsius. As indicated by the figure on the right, conventional microplate readers will stay about 3 to 4 degrees Celsius above room temperature. This, of course, also means that if the room temperature fluctuates over the day, the reader temperature will follow accordingly. However, as you can see indicated by the purple curve on the bottom here, the Ferrostar FSX equipped with AAS is able to maintain a stable temperature of 21 degrees Celsius over the whole day. This allows all plates in a screening campaign to be measured under the same conditions. It gives reproducible results across labs in different locations and environments worldwide, consistent environments for temperature-sensitive applications and kinetic measurements, and also reduces sample evaporation. The last feature I would like to highlight today is the easy automation that is available with the Ferrostar FSX. This is achieved by 3 integrated barcode readers, integration capabilities into all leading robotic platforms, multi-user control, digital signatures, FDA 21 CFR Part 11 compliance, and of course the compatibility with BMG LabTech's microplate stacker. With this, I'm at the end of my part of the talk. Martin, we are now looking forward to your presentation. The floor is yours.
First of all, thanks a lot to BMG LabTech for giving me the opportunity to give this talk. My name is Martin Schwalm. I'm from the Structural Genomics Consortium node in Frankfurt. My work focuses on degraders and PROTACs, and therefore I want to give a presentation about live-cell systems for degrader evaluation. The first question is, what are degraders in comparison to classical inhibitors? And for this I compiled this slide. So we can first have a look at the so-called occupation-driven inhibition, which is the mechanism which is exploited by most of the FDA-approved drugs. Here we have a certain protein of interest. In this case, I depicted a kinase. A kinase carries out a phosphor transfer to its substrate. And while using an orthosteric inhibitor, in this case it would be a kinase inhibitor, we would block this active site and actually stop the substrate phosphorylation upon treatment with this inhibitor. So if we now have a look at multi-domain complexes or multi-protein complexes, we see that these complexes can have more than one domain. While one of these domains carries out the active function that can be blocked by the orthosteric inhibitor, all other functions of this multi-domain complex or this protein complex will remain intact. So if we now look at target degradation approaches, we can see that we have the same protein of interest, but this time we treat it with a so-called degrader. And this degrader leads to its subsequent degradation and therefore also to the depletion of the active site, which leads to a lack of this phosphorylation event. Now, looking at multi-domain proteins or multi-protein complexes, we can see that degrader treatment binds to the same site as this orthosteric inhibitor, but it leads to the degradation of the whole complex and therefore depletes multiple functions that were carried out by this. So now we can check how these degraders are designed. So we have two main classes. One are called molecular glues and the other class is called bivalent degraders. So molecular glues are relatively small molecules. I depicted probably the most prominent one. It's called thalidomide. It's pretty much known to be the main ingredient in Contagan, but in 2010 it was found that it actually binds an E3 ligase called cereblon and mediates induced degradation of certain transcription factors. So as you might see, it's pretty hard to design these since it's not really rational. Therefore, the bivalent degrader approach was carried out. In this bivalent degrader approach, you have simply two target ligands. So one is here, one is here. You chemically link both of these, and this leads to subsequent proximity induction of two proteins. So if your molecular glue shows enough affinity towards the E3 ligase, you can also use this for the synthesis of bivalent compounds. So if you recruit two of these proteins together, the important part is that one of the proteins needs to be an E3 ligase and the other one your target protein. So in this case, the ligand recruits VHL, while this ligand would recruit cereblon. So why is this important? For this, we need to have a look at the ubiquitin proteasomal system. It's a multi-step process which ultimately leads to degradation of a certain protein. So it starts with a monoubiquitin. Monoubiquitin gets activated by this E1 using ATP. It gets transferred to an E2 where it covalently binds to this protein, and the E2 gets recruited to an E3 ligase complex. So these complexes can have different shapes and different flavors. In this case, I depicted a Cullen E3 ligase. Cullen E3 ligases comprise a backbone, the Cullen backbone, a substrate receptor, and the ring box protein which recruits the E2 enzyme. So in this assembled state, a substrate can bind the substrate receptor as depicted here, and the E2 transfers its ubiquitin onto the substrate. After approximately 4 ubiquitins forming a polyubiquitin chain, the substrate is marked for proteasomal degradation and subsequently degraded. So if we now treat the cells with a degrader, we actually interfere with this step where normally the substrate would be recruited, but due to the induction of proximity to a neo-substrate, as shown before, we recruit our protein of interest and mimic this cascade to ultimately degrade it. So let's have a look at the relevance of degraders in the clinic. So we have this time frame that was taken from a really good review, and this time frame starts at 2001. 2001 was where the Craig Cruz lab showed with the prototype system that they were able to degrade MetAP2 by recruiting the E3 ligase BTRC using a peptide. Later in that time, the first small molecules were established binding to an E3 ligase and also used for the synthesis of bivalent degraders. So in 2019, the first bivalent degraders entered the clinic and are now undergoing different clinical phases. So if we have a further look at these clinical trials, we see that different big pharmas actually are involved in these clinical trials, but the important and interesting thing is the E3 ligase that is recruited. So in this case, we see that cereblon is most of the time the recruited E3 ligase. So this has certain reasons. One of the reasons is that cereblon seems to be the most potent E3 ligase that was targeted so far. So this was actually a lucky shot since thalidomide was not intended to be a cereblon binder. But the second reason is since thalidomide is pretty much known in clinical trials, we know what to expect, what off-target effects to expect, and therefore it's not as unknown as, for example, VHL ligands in the clinical environment. So if we now look at this picture, it shows in a pretty nice way why it might be important to search for new E3 ligases. So if we look at this part, we see that cereblon, VHL, and for example, the lately drugged GIT4 are ubiquitously expressed E3 ligases. This means that they are relatively evenly expressed in the human body, and treatment with PROTACs would lead to severe off-targets in healthy tissue, while having a not really high activity in, for example, tumor tissue. Using tumor-enriched E3 ligases, on the other hand, would significantly increase the activity of our degraders in this tumor tissue, while leaving the healthy tissue more or less untouched and only exploiting the inhibitory effects of our degrader molecules. So now the important question is, how can we actually measure this small molecule-induced degradation? Since you see it's a multi-step process, it might be quite complicated. So, and I took this scheme you just saw for the ubiquitin proteasomal system, and I put it into a circle. And the reason why I put it into a circle is that degraders exhibit a so-called catalytic mode of action. So this works that we have our degrader, and this one forms a binary complex with either the E3 ligase or the protein of interest. Here I depicted the protein of interest. So after forming this binary complex, it recruits the second binding partner, which in this case is the E3 ligase, forming the ternary complex. This ternary complex formation leads to ubiquitin transfer onto our substrate, polyubiquitinylation, and degradation. Since this degradation event leads to depletion of the binding site, the PROTAC actually gets released back into the cytosol to undergo multiple catalytic cycles. So how do we measure this in vitro? We can measure binary complex formation and binary complexes using SBR. The binary complexes can additionally be measured using ITC or microscale thermophoresis. Ternary complexes more or less use the same measurement principles. For ternary complexes, due to the increase of the complex mass, we can actually use mass photometry. Mass spectrometry can be used to measure the increase of mass upon ubiquitin transfer, while the final degradation step cannot be monitored in vitro. And the worst part is that the whole process is not entirely high throughput screening compatible. As you can see from this card also is that you would need at least 2 expensive devices and multiple assay setups to assay the whole cascade until the point where you can't measure the final degradation. So this sparked the idea to actually establish a pipeline which is not only complete, but it's also working in a plate reader, and it's using live cells for these assays. And to do this, we started establishing so-called nanobread. It's a target engagement method where you can measure the formation and the affinity of binary complexes. We can use the halotech-based protein-protein interaction nanobread systems for ternary complex formation and affinity. We can use the same system by taking ubiquitin to measure the ubiquitin transfer onto the substrate, and we can use the hybrid technology to actually measure the final degradation in living cells. And the best thing is, I personally run all of these assays in our Ferrastar. I could successfully downscale all of these assays into 1536 well plates with C prime values higher 0.5, and all of them are seen to be HTS compatible assays. So to show you in a schematic way what's going on in these assays, so we can start with the so-called nanoluciferase. So the nanoluciferase can be built out of 2 parts. This one is called the nanobit system. So we have 2 bits, and these 2 bits form the nanoluciferase. So with this system alone, we are able to measure binary complexes and protein degradation in living cells. using a tracer, this also enables us to measure nanobread, and using the halotech together with the halotech ligand, we can also measure ternary complexes and ubiquitin transfer. So in principle, this system would also allow imaging, but since it's not done in a plate reader, it's not covered in this talk. So as you might have seen that these molecules have quite a large size, you might think that they might have problems actually entering the cellular system. And this is one of the main problems during degrader evaluation, that you actually don't know if the degrader is just weak or if it's not going into cells. And for this, we found that membrane passage is an extremely important parameter to measure and to calculate, to understand what are the doses that you really need to treat your cells with. So for this, we use the nanobread system actually, to compare the IC50s we generated using intact cells with IC50s we generated from lysed cells. How is this done? To do so, we, as I said, use the nanobread system. So using the nanobread system in the Ferrostar, we can use a 384 well plate, which allows us to measure up to 16 compounds in technical replicates on one plate, including all controls. We transfect our cells with an luciferase tagged full-length protein, which is subsequently expressed by the cells. So if we now use these cells and treat them with a tracer, which is shown here, we can measure the tracer binding to the protein of interest, since the dye which is attached to the tracer gets in proximity of the nanoluciferase. So we can measure the bread signal which is generated by this proximity. If we now treat the cells with our PROTACs and they enter the cellular system, we have a competition of the tracer and the PROTAC to this binding site, and if the PROTAC successfully binds the binding site, we will lose the bread signal, which can be measured and plotted against the dosage that was treated. So first, an important thing is, especially if you use a novel tracer, that you have to establish the assay. For this, you just titrate your tracer to the cells, which leads to this kind of increasing curve. So if you have a high potent or highly potent tracer, you will see that your curve increases and reaches some kind of plateau where your binding sites are saturated. In this case, we don't see this, which gives us the information that the affinity needs to be lower than 1 micromolar or weaker than 1 micromolar. So since we are not really sure if our tracer is still soluble using higher concentrations, we actually restrict our highest concentration to 1 micromolar, which therefore gives us these values as an assay window, and we now use 1 micromolar of the tracer and try to displace it using the PFI7 in this case, which is the unlabeled tracer parent compound. So using different concentrations gives us this sigmoidal curve, and we can see what the apparent IC50 of PFI7 is in the cellular system. So using this now in our example on the protein of interest, we can see that in intact cells, our PROTACs are relatively weak, while permeabilization of the cells leads to a shift of the IC50 of approximately 10-fold to the left, which means that our PROTACs are approximately 10-fold more potent in permeabilized cells compared to intact cells, which gives us the relative assumption that only 1/10 of the PROTAC actually enters the cellular system. So you might ask yourself where you actually get this tracer from, and due to this, we generated something called tracer database. So a tracer, as I just said, is just a target ligand chemically linked to, in this case, a nanobread 590 dye, you can also use fluorescein or other dyes, and then later used in these displacement assays. So our idea was that we actually take these tracers, which are synthesized by scientists all around the world, and not only scientists in academia, but also in the pharmaceutical industry, and we want to establish some kind of quality criteria to these assays. So if these molecules match the quality criteria, we will put them into tracer database, which means they are actually reviewed tracers, and we will provide all scientists with different informations about the assay. So how is the structure? This is shown here. Our input are different informations. We group the assays into different levels. So we have an expert and a robust assay given with the different parameters. And the database will carry out all the rest, like generation of structures and generation of plots. However, there's still the opportunity that we don't have a tracer in hand, and therefore it's quite good to have a tracer-free assay system. And in this case, we use something called BITSA or hybrid-based CETSA. CETSA is a cellular thermal shift assay, and together with BMG LabTech, we already published an application note on this. The principle is based on the thermal stabilization of a certain protein upon compound binding. So if our compound binds to our protein of interest, it might thermally stabilize this protein. So if you increase the heat, it might be more stable compared to its non-bound state. So what we saw in the past is that this thermal stabilization is in good correlation and good agreement with affinity of these compounds, and therefore we can use this to rank compounds according to their affinity, but it doesn't give us real IC50 values. So if we use this assay principle, we will get these melting curves, and this allows us to fit a melting point to our protein of interest treated with different compounds. And if you plot this, for example, in this bar diagram, you can actually see what the highest and what the lowest binders are, but you won't get any affinity. The problem in this case is that binary complex assays in general are most of the time not suitable for molecular glues. The reason is that molecular glues often only bind the complex of both proteins. So it binds to the surface and changes the surface to increase the binding of the second protein without really generating a binary complex. And due to this big reason and the high interest of molecular glues in the pharmaceutical industry, we focused on measuring ternary complexes. And if you check the literature, you can actually see that a lot of paper group around the ternary complex when it comes to some kind of rate limiting step. And the publication where I established these assays is also grouping here, so we are all in agreement that the ternary complex directly correlates with the final degradation. So it might actually make sense to screen ternary complexes instead of, for example, screening final degradation through Western blot. So I established this assay cascade. You can use your protein of interest, which is halotagged, and an E3 ligase, which is nanologue tagged, or vice versa. You can treat the cells with a halotag ligand containing a fluorophore, and if you bring these two bread pairs into proximity, they will emit light at this certain 590 nanometers, while washing the PROTACs out again will lead to a dissociation of this ternary complex and therefore a lack of the spread signal. So exemplary data is shown down here. Just measuring endpoint assays using this setup, we can see that we can measure affinity of our ternary complexes. measuring time-resolved association after addition of these degraders. We can measure association curves, and preparing or carrying out washout experiments, we can actually measure dissociation curves. And the good thing here is that this assay setup is suitable for molecular glue screening. And the good thing is that you don't need a stably transfected cell line or a CRISPR-Cas engineered cell line. So on the other hand, what you need is a total of 8 different plasmids. So it's N and C terminally tagged N-look, E3 ligase, or protein of interest, and an N and C terminally tagged E3 ligase and protein of interest with the halo tag. Subsequently, you have to try all of the different combinations which are possible to find the closest conformation between both bread partners. So I've shown this here in a schematic way. So if you get the highest bread signal, both partners are in the highest proximity, and therefore you get the best assay window. So down here, I've shown experimental data that I actually collected. There you can see that it's much less beautiful compared to a schematic view here. But what you can also see is that a C-terminally nanologue tagged VHL together with a C-terminally nanologue tagged protein of interest gave the best assay window in this case. So we got a relative bread ratio of around 40, which is higher compared to anything else. And the importance that you actually pick the highest bread ratio in this case is that if you see the publication that we published on this, we also contain some crystal structures where we saw that our protein of interest can rotate around 90 degrees upon treatment with different PROTAC series. So if we would have not taken the highest bread signal here, we would quickly reach a pretty low bread ratio and therefore lose our assay after twist of this protein of interest. So showing again our scheme here, here's some more exemplary data. So here at T0, I added our PROTAC, put it into the ferrostar, and then continuously collected the bread signal over time. In this case, I used CO2 independent medium since our ferrostar is not supplied with CO2. After 60 minutes, I realized that I've reached some kind of plateau and washed the cells using PBS, added substrate again, put it back into the ferrostar, and we could actually measure dissociation again of the complexes. So the last and probably the most important part of this whole cascade is to measure the final degradation in living cells. So far, I think hybrid technology is the best that you can use to measure protein abundance in living cells. So how it's normally done when you check the literature, you most of the time see Western blots and proteomics. And being honest, this is a really good validation of a degrader. So you have native protein, you see depletion of the band, and then you have selectivity screening in proteomics. Sadly, this is not the standard within the literature. And with Western blots, we have the problem of really limited dynamic range. So we can't really distinguish between these two bands. We just see that these are lower compared to these. So for this, the hybrid technology was established. So what it is we take the nanoluciferase, and this nanoluciferase is split into the so-called high bit and the large bit. The large bit is just the large part of the nanoluciferase, while the high bit is an 11 amino acid long beta barrel sheet, as shown here in cyan, and it has an extremely high affinity towards the large bit. So what you do is you take this high bit sequence and you endogenously tag your protein of interest using CRISPR-Cas9 in this case. And since you only alter your native protein with 11 amino acids, we actually saw that tagging your protein with this doesn't really change anything in the protein behavior. So the trafficking remained the same, and the cellular localization, for example, also remained the same. So this is probably the least invasive method that you can use to measure protein abundance in living cells. So having now a look at how we actually measure this is we have our endogenously hybrid tagged cell line, and we use a large bit expression vector which is transfected into the cell line. So we don't really continuously express the large bit, we just transfect it into the cells when we really need it. So what's going on in the cell is we have our protein of interest carrying the hybrid. After transfection with the expression vector, the cell expresses the large bit itself, so it complements inside of the cell in these living cells, and after addition of substrate, you can measure a signal at 450 nanometers. So treatment of these cells now with PROTACs will lead to polyubiquitinylation and subsequent degradation of our protein of interest, and therefore also of the hybrid tag or maybe the whole complemented nanoluciferase. So therefore we will see a decrease in luminescence. If you now check the data that I measured using this, so we have a negative control compound, and you can see that it indeed was pretty negative. Using active PROTACs, we can see that over time, so in this case it was 24 hours, we see a decrease in target protein abundance, and it was a really steady signal. So the question was now, what happens after 24 hours? So I tried to push it a bit further and used MS67, which was the most active PROTAC, and measured it over 60 hours. And there you can see that the first 24 hours pretty much align, so it was highly reproducible. But after 24 hours, nothing really changed in the protein abundance, which tells us that this PROTAC was highly active in suppressing the cell's ability to actually resynthesize WDR5 or our protein of interest. So the last and really cool thing that you can do with these kinetics that you measure over time is that you can use different concentrations, leading to a high variety of curves that you measure in dependence of the concentration you use. So using this principle, we can now pick any time point that we want and use it for DC50 determination. So why is this important? I mean, you could also do it by Western blot. You just take samples after 4 hours, and after 24 hours, you run your Western blot and you can calculate your DC50. But the problem was shown by Promega scientists, which compared different bed bromodomain degraders in this continuously measured system. And here you could see that BRD2 was actually highly responsive upon PROTAC treatment. So after 4 hours, you could see that different dosage induced high degradation of BRD2, while after approximately 15 hours, the resynthesis rate was so high that after 24 hours, BRD2 reached again 100% of protein abundance, where you can see for BRD4 the protein was still down. So just imagine measuring a Western blot after 24 hours, you would have assumed that this PROTAC is inactive while it actually was active. So this highlights the importance of kinetic measurements in degrader design. So to summarize all of this, I want to show you what I've learned and how I changed my screening workflow. So I call the old one the classical workflow, and you still see it in the majority of publications, and this has several reasons. So we start with screening of binary complexes, we characterize ternary complexes, and most of the time we end with a validation of degradation. So the reason is that binary complex screening is pretty easy. We have lots of assays available and it's HTS compatible. So we can screen in high throughput until we find our high affinity binder. Next, we can use ITC or MST, which has pretty low throughput to measure ternary complexes. And in the end, we use Western blot or proteomics, which also has pretty limited throughput for our degradation validation. So, and what I learned in the publication, which was published in Cell Chemical Biology, is that some PROTACs can actually show some molecular glue-like properties, meaning that they don't really show a binary complex while they still induce ternary complexes and target degradation. And using this classical workflow, I would have overseen these PROTACs. So therefore, I adapted this and I call it the optimized life cell workflow, where we start screening ternary complexes. So the reason for this is that we saw a high correlation between ternary complexes and degradation, but we don't really need a CRISPR-Cas9 stably engineered cell line. We just need to transfect two plasmids into cells, and therefore we have a highly adaptable system, and we can change our target protein, we can change our E3 ligase, and it's just flexible and much easier to adapt to your certain needs. While creation of a CRISPR-Cas engineered cell line takes multiple months. So next is we validate our degradation using the hybrid life cell system. And last, we can validate the whole assay cascade by, for example, screening binary complexes and understand why certain degraders are better than others. And with this, I want to end this presentation. Thanks again to BMG LabTech to giving me this opportunity. This is the group I work for. This is Stefan Knapp, my boss. And thanks a lot.